Hello everyone. It's a great pleasure for me to introduce to you our work and talk about biomanufacturing and intelligent control systems. First, I want to thank the International BioDesign Research Conference Committee for inviting me to give this lecture. My lab had been moving from University of Maryland to Technion, Guangdong. It's a branch campus of Israel Institute of Technology. I'm quite happy to join this new position. So as a chemical engineer, we look at the cell as the microbial cell factory. So the nice thing about the biomanufacturing is that you have flexible substrate, you have renewable, and it's low cost. In terms of enzyme, it's very specific and could be very efficient and perform a function under ambient conditions. And we can make a range of different products, including food, pharmaceuticals, drugs. But if you look specifically inside the cell, you will find an interlinked metabolic network. We call it metabolic maps or metabolic pathways. You have feedstock processing units, which is a glycolysis pathway, which can break down, sugars into mono sugars or glucose. You have a powerhouse, which is the mitochondria, to supply ATP to sustain the life of the cell. You also have the assembly line. So the cell can synthesize protein from amino acid to synthesize cell membrane from fatty acid. Another thing we cannot neglect is that the cell needs lots of reducing equivalents. We call the hydrogen plant. It's equivalent to a hydrogen plant in a chemical plant because the cell needs to deposit electrons to build carbon-carbon elongation chemistry. As a metabolic engineer, we normally look three golden index, that the titer, yield, and productivity, to evaluate the final performance of the strain. We start looking for genome or searching for genomes for valuable metabolic pathways or maybe a gene cluster including a functional metabolic pathway which can give us deliver some valuable compounds or drugs or maybe some interesting molecules. Then we do codon optimization, then we stitch the gene together with the Gibson assembly, we can make an entire metabolic pathway. Then we can iterate and test stream performance. If we found there's some missing components, there's feedback inhibition or regulation, we can remove the, for example, phosphorylation set or maybe post-translational modification. We look at the protein engineering to improve the efficiency or maybe the specificity of the enzyme, so we can keep iterating the process until we reach our target yield or productivity. Then we put the upstream in the bioreactor, we scale up and we test the performance of the strain. Eventually, we may be able to reach the society, maybe economic impact. This is the entire process of biomanufacturing, but I want to see the downstream purification, the processing is very critical at determining the final cost of the products, whether they can be achieved in the final market or not. So here's another rhetorical comparison between a chemical plant and a real microbial cell factory. On the left-hand side, you can see if you have a central computer in the chemical plant, we can use this C language and maybe Python to program this 0101. Eventually, we can control the mass flow, energy flow by controlling the valves or maybe the efficiency of the flow rate of the heat exchanging process and maybe the cooling process. But on the right-hand side, the cell is a very intelligent system. As a synthetic biologist, what we do actually, the central computer is the CPU of the cell, is the blueprint, is the genome of the cell. They send the instruction to the cell, but as a synthetic biologist, actually we can design and engineer genetic application apps. So in such a way, we can insert these genetic apps into the cell. Then this genetic language will be decoded and will be interpreted and executed by the genome. Eventually, they can output different functions. In this meaning, we can reprogram the information flow in a cell and control the mass and energy to maximize the biomanufacturing efficiency. So in today's lecture, I want to give you a flavor how we are going to transition from a rational engineering approach to intelligent control systems. So the first example primarily will be the stoichiometric based mass balance. So I'm going to use the yin and the yang balance to improve the lipid production, and the bridge or the connecting point between rational engineering and the intelligent control will be the biosensors, especially as the transcriptional factor-based biosensors, which allows us to precisely probe the cellular states. And the last point will be the intelligent control system. With all the two, approach converge together, we think, at this point, we'll be able to encode the decision-making function, encode complex logic function into cells, use the interlinked feedback genetic circuit, so we can make the cell dynamically respond to their changing environment to adjust their, growth fitness to improve the biomanufacturing efficiency. So the first topic about the rational engineering, I want to talk about the palm oil challenge, palm oil process. So around 80 to 90% of the palm oil was manufactured in the Southeast Asia, Indonesia, and Malaysia. So in the recent years, we see the world has a growing appetite for palm oil. For example, this year we have around 77 million metric ton demand. It's primarily used in food and beverage, biofuel, energy, and industrial, maybe personal care, cosmetic industry. So the palm oil is associated with lots of environmental concerns because in recent years we found there's a massive scale deforestation in the tropical area. For example, 80 to 90% of the palm oil were manufactured and planted, supplied in Southeast Asia, primarily Indonesia and Malaysia. So this island called Borneo Island is the largest island in Asia and the third largest island in the world. It's bigger than the size of the Texas. So around 65–75% of the tropic forest disappeared in the past 30 years because this is a major economic plan in the local place, and the people are always trying to overexploit and try to expand their palm gardens. It turns out to be a very bad choice when they try to make more money. Actually, the climate change and it getting imbalanced, rain season and the drought season. Eventually, they get the production, the quality even worse and worse year after year. There's a child labor, there's a loss of ecological diversity, and lots of negative environmental impacts. Even that our current agriculture and planting technology cannot meet the growing demands of the palm oil industry. Our world has seen a sustained appetite demanding for the palm oil industry. So we see this oleaginous yeast lipolytic could be a solution to fix this problem. So this yeast naturally make large amounts of lipids, up to 20% for the wild type strain, to all the way up to 90% in the engineered yeast. So this yeast is very robust. It can grow, on a broad range of substrates, including hexols, the glycerol, and the municipal organic waste, VFAs, and some hydrophobic, alkanes, wax, maybe some crude oil, extracts. It is also genetically tractable. So compared with other type of organism, Rhodosporidium, it's very easy to engineer, Yerba lipolytica. For application, this is a grass organism approved by FDA, can be used in the food beverage industry. It has been engineered by DuPont and DSM to produce or manufacture, polyunsaturated fatty acids such as EPA and DHA as the infant formula supplements. Of course, it can be also used for personal care, cosmetic to make this shampoo and some, special shea butters in the commodity chemical industry. So why this yeast is so good at making lipids? So if you check this yeast, we know this belongs to the eukaryotic family. They have different cellular compartments, the mitochondria, peroxisome, and the lipid bodies, Golgi bodies. So this cellular compartment allows this yeast has dedicated acetyl-CoA and malonyl-CoA pathway. Another very important thing is that some of the genes or the proteins, especially for the enzyme responsible for fatty acid synthesis, such as acetyl-CoA carboxylase fatty acid synthesis and acyl-CoA transferase, they are co-localized as the gene as a protein cluster. So they can very efficiently channel the precursors, the acyl-CoAs, to all the way to the DAG, the diacylglycerol and the triacylglycerol, at this interface between ER and peroxisome. They can facilitate the mass transfer and the lipid particle aggregation very efficiently. Another nice thing about this yeast, there's no Crabtree effects. If you feed the yeast with a large amount of glucose, they won't make ethanol at all. They will just make lipids like crazy. This is the nice thing about its yeast. That's why we call it a lipo yeast. So if we check the fatty acids biosynthetic pathway, you will find there's a two-step which is highly demanding for reducing equivalents in the form of NADPH. The first step we call beta-keto isoACP reductase. So you need the electrons to reduce the ketone group to a hydroxy group. The second step will be the eno-isoACP reductase. So you can reduce the saturated double bond to a unsaturated double bond to a saturated, carbon-carbon backbone. So every 2 carbon elongated, incorporated, you need 2 NADPH as a reducing molecule to have the chemistry run efficiently in this cycle. So here is the detailed analysis of stearic acid biosynthetic pathway. Has contained 18 carbons. All the 18 carbons has to come from the backbone oxidation of glucose. It comes from acetyl-CoA and malonyl-CoA. We know because for the glycolysis pathway, 1/3 of the carbon was lost. In the end, the 18 carbon actually come from 27 carbon atom, with corresponding to 4.5 mole of glucose. As we know, to synthesize stearic acid, you need 8 steps of the chain elongation cycle. It requires 60 molar of NADPH. All these 60 molar NADPH actually come from the splitting of additional 1.33 molar of glucose. As a result, overall mass balance you will need 5.83 molar of glucose to synthesize 1 stearic acid. So the lipid sugar conversion yield could be quite low. It's around 0.271 gram per gram. Here I want to have another, metaphor we call the yin and yang balance. For the yang, basically we call acetyl-CoA and malonyl-CoA the yang because they contain the ketone group. They like to combine with the electrons, so they are electrophilic. For the yin, they like to combine with the ketones, the eno groups. They are basically nucleophilic. When you keep the yin and the yang well balanced, so the fatty acid biosynthetic pathway elongation cycle can run as efficient as possible. So when we check specifically inside the cell, we do find there's an alternative pathway to generate reducing equivalent. For example, in the block 1, it's the canonical pentose fast pathway. So in terms of stoichiometry, you need 5.83. So the theoretical yield is quite low. The sugar to lipid conversion yield is only 0.271 gram per gram. If you can choose alternative pathway, for example, the GABDH, if you can change the cofactor from NADH to NADPH, eventually you can reduce the glucose consumption from 5.83 to 5.08. And all the way you can go to block 3, use the POM cycle we call pyruvate oxaloacetate and S-malate cycle. So this is a transhydrogenase cycle. You can reduce glucose to 4.8. And if you go to block 4, so you will need we call the non-oxidative glycolysis pathway. This pathway actually is very efficient. They will just need 4.7 glucose to make 1 stearic acid. So the theoretical fatty acid yield will be significantly improved from 0.27 gram per gram to all the way to 0.33 gram per gram. It's around 34% of increase. So here I'm showing you the generic stoichiometric balance, the yield curve, the carbon conversion yield curve, which corresponds to the blue dots and the red dots, and the raw material cost curve, which is the golden curve. If you can plot the yield and the cost on the y-axis, as a function of the RQ, the respiratory quotient, you will find actually it represents the oxidative states of the carbons of the so in different pathway, as we mentioned in the previous slide, if you move from the pentose fast pathway corresponding to 0 point to all the way to the GAPDH corresponding to point 1 and 2 corresponding to palm cycle and 3 corresponding to the NOG pathway, you can significantly improve the carbon conversion yield. At the same time, you can see the raw material cost will be significantly driven down. As a result, at least you can save around 40% of the raw material cost. Here we can conclude that the source of the NADPH actually determines the carbon conversion efficiency and overall cost. This is quite significant if you try to run a company, operate a plant. We did validate and test different alternative NADPH pathways to try to improve the lipid yield. Eventually, we found the optimal in source and we choose the top choice pathway. It's the NADPH specific GAPDH enzyme and the palm cycle. So it's the pyruvyl, oxaloacetic acid, and malic cycle. So we grow the engineered cell into our bioreactor. We found the cell can accumulate up to 100 grams per liter of lipids, and the oil content reach around 72%, and the process yield is 0.286 gram per gram, which is already beyond the limits of the pentose fast pathway, and the productivity reach 1.2 gram per liter per hour. If you check the morphology, the shape of the cell under microscope, you see actually our cell, once we have enough NADPH, the cell actually is rounded, is kind of a single, is isolated and has a spherical structure. It's increase their contact surface to the nutrients, so they can very quickly to uptake the nutrients. As compared to our wild type cell, it will have stress and become elongated and high fat morphology. So this is quite different and significant for our experience to engineer this lipid. So the stoichiometric-based mass balance could be a very efficient strategy to choose the best metabolic routes. But as we heavily engineer the yeast, the cell got to sense their environments. There's lots of burden. There's lots of changing, condition in the cell. So we need to look how the cell will cope with the environment, how they will probe their cellular state. And in this section, I'm going to move to the biosensor topic. So the first example I want to demonstrate is that there is a light sensor based on the chemistry of the sulfur housekeeping metabolism in the cell. There's a gene called MD25, which encoding enzyme responsible for assimilating hydrogen sulfide to synthesize L-homocysteine. L-homocysteine is a direct precursor to synthesize methionine. So where does the hydrogen sulfide come from? Actually, in the cell, there's a number of reduction steps. They can reduce sulfate all the way to hydrogen sulfide. Imagine if you delete MD25 gene. So the hydrogen sulfide will be accumulated in the cell. At the same time, if you feed the cell with soluble lead, so for example, lead acetate, lead nitrate, naturally this hydrogen sulfide will be precipitated by the lead, will form the black colony called PBS, lead sulfide precipitate. It's very interesting. We found this could be a very sensitive light sensor. As you increasing light concentration in the drinking water, you can see the yeast colony become darker and darker. It can detect light concentration up to 10 ppm. So in a word, we could develop a portable, inexpensive light sensor to represent the heavy metal concentration, including we can easily expand this sensor to include cadmium or maybe silver or maybe other heavy metals to detect drinking water. So the second example I want to give you is a transcriptional factor-based biosensor. The cell actually has a very delicate controlled mechanism to get rid of hydrogen peroxide. They use two enzymes. One is called glycosylceral reductase, and the other one is glycosylceral peroxidase. Under stress of hydrogen peroxide, SKN7 YAP1, they will be first, translocated into the nucleus, and they will form a heterodimer, and they will be co-localized onto the SRE and YRE bonding sites. What happens is that the structure will change, and there is a disulfide bond, intermolecular disulfide bond will be formed, and they will sequestrate their nuclear export signal. So the two proteins will be trapped or sequestrated into the nucleus, so they can keep recruiting or activating RNA polymers. The RNA polymers actually will be responsible for transcribing the genes encoding for glycosylceral reductase and glycosylceral peroxidase. As the cell expresses enough of the peroxidase and glycosylceral reductase, you can imagine that the reduction state of the cell will get back to normal. Then the disulfide bond formed in SKN7 YAP1 will be released. They will be formed into a cell group. Then will be so this SKN7 YAP1 heterodimer will dissociate and be get exported outside to the nucleus. In this way, the cell can cope with the oxidative stress. Based on the information we mentioned above, here we develop a genetically encoded biosensor to detect the varying concentration of hydrogen peroxide. First, we place the YAP1 and SKN7 under the control of a strong constitutive promoter. Under the, we challenge the cell with the varying concentration of hydrogen peroxide from 0 millimolar as a negative control all the way up to 1.5 millimolar. And we use a hybrid promoter, we use a minimal leucine promoter, and of course we design the YRE and the SRE bonding site in front of our leucine promoter. And we place the luciferase reporter gene under the control of the leucine promoter. As we increasing the concentration of hydrogen peroxide, initially at the first 10 to 15 minutes, we do see the cell start expressing luciferase linearly, but after around 15 minutes, all the signals reach a plateau and they start decreasing. And initially, we are quite perplexing to try to interpret these results. It's just counterintuitive because we think the cell will keep, building up. The signal should be linearly increased all the way. But eventually we believe actually after we understand there is a strong, oxidative stress defense pathway, the endogenous pathway actually can help the cell cope with the hydrogen peroxide. We know there is glycosylceral peroxidase and glycosylceral reductase. So the cell actually, after 15 minutes, the endogenous protection pathway start be transcribed, and they will start bring down the hydrogen peroxide signal. Until 1 hour, we see all the luciferase signal will get back to normal. So there's another information we want to know. Actually, the yeast cell can very quickly adapt to this changing hydrogen peroxide concentration. Around 10 to 15 minutes, they can quickly take action, so they can start bring down the oxidative stress signal. In the lab, we also developed other sensors based on the pigmentation of some plant metabolic pathway. For example, shown here is the violacin pathway from the aromatic, amino acid pathway. We also formed this green colony based on the beta glucuronidase activity, and the M cherry, of course, is pink, and some other red color. So this sensor could be very important for us to probe the cellular state, and which allows us to interface or connecting the internal gene expression with the external environmental stimuli. So this kind of bridge can tell us to know or represent the cellular states. So in the next section, I want to move to the intelligent control system. In this section, I'm going to tell you we can encode decision-making function into cells. In such a way, we can consider both the evolutionary process. Also, we can think about the populational genetics. How we use, the external signal as a feedback control signal to control the growth fitness, and how we're going to screen the strain with better production or better performance in the cell. Back in 8 or 10 years ago, when I was a graduate student in Professor Matthew's Kopa's lab at RPI, I was working on the melanoid-CoA projects. Our aim is trying to maximize fatty acid production. But we know melanoid-CoA is an essential precursor for fatty acid biosynthetic pathway. At the same time, when we have too much melanoid-CoA, we found the melanoid-CoA will start inhibiting cell growth. So at that time, we think a possible solution that we can use a dynamic approach to engineer a melanoid-CoA switch. In such a way, we can autonomously control the level of melanoid-CoA inside the cell. When we dig into the literature, we do find that the dual transcriptional factor, they can either behave as an activator or behave like a repressor, depending on the upstream activation sequence, or maybe the fiber bonding sites. When they attach to different promoter, they will behave very differently. So to design the switch, we place the melanoid-CoA source pathway ACC under the control of a PGAP promoter. At the same time, we place the FS, the melanoid-CoA sync pathway, under the control of the T7 promoter. When we test the cell, we build the gene circles, we found the melanoid-CoA level inside the cell start oscillating. It's very interesting and perplexing, but we do publish our results on PNAS in 2014. A few years later, when I started my independent research group at the University of Maryland, I started thinking about what happened with this dynamic system. What's the design criteria we can build a perfect genetic switch? Then we start building mathematical models. For example, here, the first equation tells us it's taking the reversed form of Monod equation. We have the inhibition term, K1, under the denominator is specifically mentioned the melanoid-CoA. We have the third equation, the rate change of the fatty acid synthesis pathway actually is down-regulated by FAPA. It's taking the mathematical form of a Hill equation. At the same time, we have alpha-2 time. It's the growth-associated expression of FS because FS is an essential gene. We cannot ignore, the growth-associated gene expression. Similarly, we're taking the rate change of ACC, taking the form of the inverse, the form of the Hill equation form, because FAPA was supposed to upregulating or maybe activate the expression of ACC. In the same way, we have alpha-3 mu x, which denotes or represents the growth-associated expression of ACC. Similarly, because ACC is the essential gene for cell growth. When we model, when we design, when we assign the parameter, we found at different conditions the fatty acid biosynthetic capacity of the system behaves very differently. It could vary from from 2 grams per liter up to 12 grams per liter, up to more than 6-fold or maybe 10-fold change. When we check the maximum fatty acid biosynthetic capacity, we found the system got to be oscillating like a perfect oscillator. If you check here, the system shows here the blue curve. Actually, the perfect oscillation corresponding to the fifth panel. If you see the melanoid-CoA as a perfect oscillator. So physically, we think this system could like a spring, as physically, if you press the spring, they store the potential energy, convert velocity and the mechanical energy into a potential energy. If there's no friction, so the spring, the motion of the spring will behave like a perfect oscillation. In the same way, we believe our melanoid-CoA switch under this condition, they will behave like a perfect oscillation. So there's no friction, which means they can dynamically compensate the enzyme activity for ACC and FAS. But in our real experiments, actually we found it behaves like a damped oscillation because there's always protein degradation. There's always other factor affecting the cell growth and affecting the gene expression. So the second intelligent control system, actually we can apply them to mitigate competing metabolic flux. At the same time, we can stabilize long-term stream performance. I'm going to give you one example to address some of the challenge with the bioproduction or biomanufacturing. For example, here, we try to produce flavonoids from the same yeast, Yerba lipolytica. We know this yeast will make lots of fatty acids, but we cannot knock out the fatty acid biosynthetic pathway. We know fatty acid is essential for cell membrane biosynthesis. So how do we deal with this competing metabolic flux, because both flavonoid and fatty acid derive from malonyl-CoA? The second challenge is that as we express too much of the heterologous gene in the plant biosynthetic pathway, we found the cell has to experience lots of metabolic burden. As we grow the cell, we found the cell can regain their growth fitness, but the flavonoid production will become decreasing. If we can sequence the mutants of cell, or maybe we can check the morphology, we found the strain will be degenerated. Or we have to, the question comes here: how can we use intelligent control system to resist this strain or to prevent the strain degeneration? To solve the challenge, we have designed and engineered a bilayer dynamic control system to mitigate the carbon flux competition. At the same time, we try to stabilize the long-term stream performance. For example, here in the layer 1, which corresponds to the upper left panel of the block of the box. So the layer 1, we use the negative autoregulation circuits. When the cell starts building or making up too much fatty acid, we have a transcriptional factor endogenous protein called POR1P. They will be activated, and this protein will start driving the expression of the guide RNA. This guide RNA will bind with the D-Cas9 with the catalytically dead Cas9, so they can search on the genome. They will find the PAM sequence, but they won't cut or cleavage the DNA. But they will function as a transcriptional repressor, so they can feedback inhibit the expression of fatty acid synthesis. So in such a way, when the cell senses enough fatty acid sensed by POR1P, so they will shut down expression of fatty acid synthesis. In the second layer, actually, we build another positive feedback control system. What happened is that we found there is another sensor called FDR. They can be activated by our final product, the flavonoid naregene. In the presence of a high concentration of naregene will activate the FDR. Then FDR can drive the expression of an essential gene of leucine, LEU2 gene, which is essential for cell growth. Of course, on the genome, on the background of the cell, we have to delete the LEU2 gene. So the LEU gene has to be exclusively expressed under the control of the hybrid, PFDO type promoter. It's only be activated in the presence of naregene. So we use this linked positive feedback control and auto-negative regulation control. We hope to mitigate carbon flux competition. At the same time, we try to stabilize the long-term stream performance. To achieve our goal of bilayer dynamic control, we need to build two sensors. One is the POR1P based fatty acid sensor. The other one will be the FDR based flavonoid sensor. So we characterize a number of hybrid promoters. We place the POR1P bonding sites, a different combination of POR1P bonding sites in front of our core promoter. We do find eventually there's a combinatorial bonding site called A1, R1, A1, the A3, DNA bonding site will be most efficiently to have the broad dynamic range up to 24 gene expression change and can respond oleic acid up to 5 grams per liter. At the same time, when we test the FDR, we use the minimal type promoter with of course the FDR bonding sites. Eventually, we found only in the presence of naregene the cell can grow, because naregene can turn on the expression of the LEU2 gene expression. In the absence of non-regening, the cell won't be able to grow on the agar. So in this case, we have achieved the tight control of the leucine biosynthetic pathway, which allow the cell to be addicted to non-regening. Only non-regening will confer the cell better growth fitness. So we can log in another way, equivalent to say we log the flavonoid production with cell growth fitness through this essential amino acid leucine. So I skip lots of results. We build at the intermediate step, we build lots of the CRISPR-Cas9. We try to test the combinatorial guide RNA, which guide RNA sequence is the most efficient to feedback regulate the fatty acid biosynthetic pathway. We also design, to match the dynamic of the negative autoregulation circle and the positive, addiction circuits. Eventually, when we couple the metabolic addiction with the negative autoregulation, we test our gene circuits, this string, the bilayer dynamic control string with a control string. We do found, the wild type string as we gradually passaging and passing the cell from, up to 300 generation, the cell will gradually regaining their growth fitness. At the same time, we found that they will rapidly starting decreasing the flavonoid production without this dynamic bilayer dynamic control. For our link feedback control genetic system, we found the cell can behave quite robustly up to 300 generations. They still can maintain around 91% of the non-regening production. So our conclusion is that this growth coupled metabolic addiction can stabilize the long-term strain performance and can reduce the non-genetic strain to strain variation. So this result was published actually last summer in the journal Metabolic Engineering. So in the next couple of slides, I want to mention why it's so important to build the growth-linked phenotype, to build the metabolic addiction phenotype. Suppose we have an engineered cell, we will undergo a retro mutants as a frequency of theta. At the same time, only our engineered cell will produce our final product in the growth-associated and non-growth-associated term alpha and beta. At the same time, our final product will metabolically coupled with our engineered cell. Either it could be addiction or maybe causing a metabolic burden through the coefficient we call gamma. If you check our metabolic, growth fitness function, the mu1, which corresponding to the engineered cell, we have a gamma term. So the gamma is the metabolic coupling coefficient. When the gamma is positive, which means our final product will be benefits the growth of the engineered cell. If the gamma is negative, which means the final product will be metabolic burden to the cell. But generally, gamma will be negative, and the mu2 is not associated with any gamma. Because there's a retro mutant, there's a frequency of theta, there's a retro mutation efficiency of frequency. We can build the mass balance for the engineered cell X1 and the retro mutant cell X2. Also, we use the growth-associated, non-growth-associated production. We also build the substrate consumption balance. For example, the related with both growth-associated and non-growth-associated. Also, they related with the product formation. Eventually, we know that theta is a retro mutation frequency. It can only take the value from 0 to 1, and gamma will be a metabolic coupling coefficient. Ideally, it will take the value from -1 to +1. And we take a different parameter called delta. So delta basically is the population of the mutant cell relative to the total population of the engineered cell and the mutant cell. So the delta will be a function of theta and gamma. So we define gamma will be a degeneration frequency. We next assign the different values and try to evaluate how strong the metabolic stress or metabolic addiction will affect the stream performance for the long-term stream performance. We model the system in the CSTR because in such a way we can change the growth fitness, because only under steady state the non performing cell or maybe the less growth fitness cell will be washed out from the will be not selected by the CSTR. What happens that on this system, we found if you have a negative growth coupling coefficient, the CSTR only retain the cell for the degenerated cell. For example, here the degeneration efficiency will up to around 80 or 90% when the dilution rate reached 0.8. In the same way, if you have the positive, gamma, which means the final product benefiting the growth fitness, the degeneration efficiency will be kept down under 4%. For example, for the gamma 0.1 and gamma 0.2. On the other way, if you see the relative production increase, you will see actually when you have a metabolic addiction, when you increase the dilution rate, actually the production can increase quite significantly. At the at the dilution rate 0.8, it can increase up to 40% and all the way up to around 50% and 80%. At the same time, if you have a metabolic burden for the negative gamma, you can see actually the cell will perform very bad. Around 75 or 80% of the cell, the production will be lost. So the reason we build this model because we want to emphasize the metabolic addiction is a very important thing if you can link your final product to the growth fitness. In the CSTR, you can selectively enrich the cell which perform or which grows better, which correspondingly can perform better, making more products. In the end, I want to summarize, we have go through a transition from a rational engineering approach so we can control the mass flow based on stoichiometry based mass balance. Eventually, so as we going on, we understand the biosensors are very important, and we can use the transcriptional regulation as a lever, so we can leverage the transcriptional based biosensors to link the gene expression with the external environmental stimuli. Eventually, we can, based on the artificial intelligence, or maybe we can build intelligent control system so we can switch on and switch off gene expression and achieve autonomous gene expression control in the system. At the end, I want to thank for my previous advisors, my mentors at the RPI and MIT, and my collaborators at the UMBC, and some of my collaborators and my mentors around the UIUC, Delaware, WashU. Thank you for your mentoring and help. And also I want to thank our founding agency, Bill and Melinda Gates Foundation, the National Science Foundation, and also some of the publishers like BNB and elsewhere. Thank you for your time, and I'll be happy to take your question offline because this will be a virtual discussion here. Thank you for your time.